Hello and welcome. I'm Gwen Taylor, Senior Developmental Editor with Current Protocols at John Wiley & Sons, and I'm delighted to introduce today's webinar titled Cancer Stem Cells and Mechanisms of Multidrug Resistance by Flow Cytometry. This webinar is being co-sponsored by Current Protocols and Thermo Fisher Scientific. Thermo Fisher Scientific is the world leader in serving science. Their mission is to enable their customers to make the world healthier, cleaner, and safer. They help their customers accelerate life sciences research, solve complex analytical challenges, improve patient diagnostics, and increase laboratory productivity. Through their premier brands, Thermo Scientific, Applied Biosystems, Invitrogen, Fisher Scientific, and Unity Lab Services, they offer an unmatched combination of innovative technologies, purchasing convenience, and comprehensive support. Current Protocols is in its 29th year and is the largest collection of peer-reviewed, authoritative, and regularly updated step-by-step research protocols available for life scientists worldwide. Now with 18 titles and over 17,000 protocols, Current Protocols is part of Wiley Publishers. During today's program, we encourage you to submit your questions throughout the event by clicking on the Ask a Question box at the bottom of your screen. Your questions will not be seen by any of the other attendees. The webinar will be recorded and available for viewing in the next few days, and we will send you an email with details on how to access the on-demand webinar, along with a customizable certificate of attendance. So now it is my pleasure to introduce today's speaker. Dr. Jordi Petriz received his BSc degree in biochemistry and animal biology from the University of Barcelona, and then pursued his PhD at Barcelona in physiology and immunology, specializing in functional-based mechanisms of multidrug resistance against anti-cancer agents. Since 2000, he has been a member of the Subcommittee on Quality Assessment of Hematopoietic Stem Cell Grafts and is a principal investigator at the Joseph Carreras Leukemia Research Institute. Dr. Petriz has authored nearly 100 research publications, and his current work focuses on linking stemness with ABC transporters. He is studying several genes involved in different aspects of stem cell activation, including some that encode for multidrug resistance transporters and others that regulate self-renewal and differentiation. He has also been the president of the Iberian Society for Cytometry since 2007. So let's go ahead and get started with a very warm welcome to you, Dr. Petriz.
Yes, thank you very much for this nice introduction. First of all, I would like to thank Wiley and Thermo Fisher for inviting me to participate in this webinar meeting on flow cytometry. And my experience in the flow cytometry field starts in 1991, and my talk will focus on the study of stem cells mainly applied to learn more from the hematopoietic system. My PhD was in the field of functional flow cytometry and multidrug resistance, and most of this knowledge has applied to stem cell studies. More recently, we have explored the effect of cellular hypoxia on gene expression analysis. During this talk, we will see different techniques aimed to identify as well to isolate very rare stem cells and their subpopulations. In fact, CD34 stem cells from the hematopoietic system are the best studied compartment so far. In this slide, you can see how the hematopoietic stem cells, which are CD34 positives, are able to self-renew and to differentiate into progenitors of all hematopoietic lineages. And these processes are regulated by a large number of cytokines to give rise to red and white cells and platelets. More sensitive measurements of CD34 positive events should have an impact on research in a broad range of scientific and clinical disciplines, and could also clarify the number of CD34 positive cells needed for the engraftment. In this slide, we can see a no-lyse, no-wash flow cytometry technique developed in my lab aimed to study more precisely the absolute number of CD34 positive cells in analyzed whole blood that can be applied to cord blood, peripheral blood, bone marrow, and leukapheresis products. As you may know, flow cytometry is a multiparametric technique, and multiple cell characteristics can be studied at a single cell level. Some dyes allow us exploring the function, such as the mitochondrial membrane potential. In this slide, we can see how the mitochondrial membrane potential of viable CD34 positive cells stained with fluoramine 1-3 helps to distinguish the primitive compartment within the hematopoietic stem cells with the lowest mitochondrial membrane potential, as these cells are mainly quiescent. In addition, CD34 positive enriched cells from human bone marrow can be used for additional immunophenotyping experiments. The composition of CD34 positive subpopulation differs between bone marrow, blood, and cord blood, and in this slide we can see highly enriched CD34 positive cells that were stained again with CD38, a marker classically used to distinguish the more primitive fractions within the human hematopoietic stem cell compartment. In this slide, we can see a representative CD34 positive versus dye-cycle violet measurement in peripheral blood and marrow cells using flow cytometry. Dye-cycle violet is a DNA selective and cell membrane permeant stain which effectively subsets and orders CD34 positive cells for the definition of different functional properties that can be related to the characterization, resolution, and purification of primitive hematopoietic stem cells in combination with a specific useful marker for multicolor flow cytometry measurements. The first dot plot on the left displays normal bone marrow and malignant disease of the bone marrow with low CD34 expression, acute myeloid leukemia cells on the right. Moreover, we can use a series of monoclonal antibodies against CD34 positive subsets, and in this slide we can see CD34, CD38, and DR positive cells that were sorted and stained with propylium iodide for cell cycle studies, showing that a large number of cells were in the G0, G1 phase and the rest near the 15% under proliferation. This slide displays the cell cycle analysis of CD34 positive, CD34 positive, DR negative sorted cells, showing that almost all cells were quiescent, suggesting a more primitive enrichment of hematopoietic stem cells. And this slide, and at the same time, CD34 positive, CD30 negative, DR negative sorted cells showed that most of cells were more quiescent, giving support to the hypothesis that the more primitive compartment is enriched in G0 cells. However, in order to distinguish the G0 from the G1 phase, different studies are needed. In this slide, we can see CD34 positive purified cells from peripheral blood that were incubated with Hedgehog 33342 for DNA staining and pyrogen Y for RNA staining. As expected, most of cells are distributed in the G0, G1 fraction, but pyrogen Y dim cells or RNA low cells are enriched in G0 cells. Thus, the lack of specific markers for stem cells makes the physical identification and isolation of this compartment difficult. Since hematopoietic stem cells differ in repopulating and self-renewal potential, we also studied their telomere length by flow fish, showing that CD34 positive cells revealed remarkable telomere length heterogeneity, with a hybridization pattern consistent with different classes of human hematopoietic stem cells, suggesting that cells with large telomeres could be enriched in long-term repopulating cells. However, to complicate the issue, there is experimental evidence demonstrating the existence of cells in the CD34 negative population of human bone marrow and cord blood that can repopulate the bone marrow of immunodeficient mice. And clearly, deciding which marrow cell population to select is a crucial and extremely important issue. Magnetic activated cell sorting has been used for large-scale positive selection of CD34 positive cells. The negative fraction is a convenient source for T cells, but is also enriched in CD34 negative stem cells. Hematopoietic stem cells can be purified based on the efflux of fluorescent dyes such as rhodamine 123 and Hedgehog 3342. Although expression of ATP binding cassette transporters has been assumed to contribute to dye efflux component of the stem cell compartment, little is known about which transporters are expressed or what function they might be conferring. Nowadays, we know that CD34 positive cells mainly express ABCB1, whereas CD34 negative stem cells express the ABCG2 transporter. Multidrug resistance has been related to decreased drug accumulation using pharmacological assays. To directly demonstrate this at the single cell level, we incubated ABCB1 EGFP transfectant pools with the fluorescent chemotherapeutic drug doxorubicin and visualized the corresponding fluorescent patterns by confocal microscopy. The representative field is shown here to reflect the inverse relationship between PGP EGFP overexpression, green fluorescence localized in the plasma membrane, and doxorubicin accumulation, nuclear red fluorescence. In this slide, we can see how these transporters contribute to dye flux. In our lab, we have developed ABCG2 transfectants that can be easily monitored by fluorescent techniques and, of course, using flow cytometry. Many drugs are good fluorescent substrates, and these fluorescent substances can intercalate into the DNA and accumulate in the nucleus. This reduced drug accumulation pattern present in the ABCG2 expressing cells can also be reversed by pre-incubation with some reversal agents, such as verapamil or cyclosporine A. Flow cytometry is increasingly used for quantitation of cellular fluorescent drug retention, effect of reversal agents, and for the expression of drug resistance-related cellular surface markers. To achieve optimal growth conditions, cells were seeded before the drug uptake and retention experiments in multi-well tissue culture plates in medium. Uptake of drugs is performed by adding them to the culture medium in the presence or absence either of verapamil or cyclosporine A or other inhibitors. The concentrations of the fluorescent substrates is previously optimized to allow appropriate electronic signal compensation. After 1 hour of incubation, cells are washed, fed with drug-free culture medium, and cultured for 1 hour at 37 degrees centigrade again in the presence or absence of the reversing agents to evaluate their effects on drug retention and monitor it on a flow cytometer. This slide describes the application of multiple substrates to identify drug-resistant cells in the green channel, such as rhodamine 123 or Cyte13, doxo and daunorubicin in the orange channel, or mitochondrion in the far red channel. Optimal staining and concentration should be optimized and combined with immunophenotyping. Gating of viable cells is based on light scatter parameters and in simultaneous staining with propylium iodide or 7-AAD. Analysis of 10,000 cells per sample is carried out in the fluorescent count log histogram, collecting the autofluorescent signal in the first decade. Cancer and leukemic cells can simultaneously express different multidrug-resistant transporters, which can be easily distinguished by combining fluorescent probes and multidrug reversal agents. Cyclosporine A is generally specific for P-glycoprotein and probenecide for ABCC1. By using calcein AM, a molecular probe that can be excluded by these two transporters, different fluorescent profiles can be observed, indicating that cells can evade anti-cancer treatment consisting of multiple drug combinations. As we can see in this slide, flow cytometry can also be used to monitor drug efflux over time. Stable ABCG2 transfectants were preloaded with mitochondrion, a fluorescent drug that can be excited with red lasers, showing that specific transporter mutations consisting of a single amino acid substitution are crucial for changes in the velocity of the transporter, making cells more resistant to treatment. As you can see here, maximum transport velocity can be easily estimated, and this information can be very helpful for clinicians because will have a special impact on a reduced drug efficacy or effect. Multidrug resistance in human leukemia and lymphoma can also be detected by using monoclonal antibodies such as MRK16 and MRK20. The immature CD34 phenotype is strongly linked with the P-glycoprotein. An important advantage of flow cytometry for the detection of multidrug resistant cells seems to be the possibility to identify multiple subsets of hematopoietic cells using lineage-specific or differentiation antigens. Those cells can be separated using positive or negative sorting procedures for gene expression analysis. Here we can see the phylogenetic tree of 3 human ABC transporter proteins. In this figure, only a part of the human ABC transporter phylogenetic tree is presented, showing members of the ABCB, ABCC, ABCG family. The transporters discussed in this webinar are ABCB1, expressed in CD34 positive cells, and ABCG2, expressed in CD34 negative site population cells. Site population, SP, has become an important hallmark for the definition of the stem cell compartment, especially for the detection of stem cells and for their physical isolation by fluorescence-activated cell sorting. SP cells are CD34 negative and were discovered using ultraviolet excitation based on the efflux of H33342, and more recently, a new assay based on the efflux of vibrant dicycled violet stain, DCV, has been documented to discriminate site population cells. Site population displays a unique low H fluorescence emission property, and H low cells are described as site population cells by virtue of their typical profiles in red versus blue vibrant dot plots. Moreover, the degree of efflux activity seems to correlate with the differentiation state. In fact, SP cells showing the highest efflux activity are the most primitive in terms of the differentiation potential. And ABCG2 is the only responsible for the formation of the H33342 flow cytometric fluorescent profile, and this profile is blocked in the presence of multidrug reversal agents because H efflux is reduced in the presence of fumitremorgine, verapamil, and other drugs. The site population displays a very interesting chromatic shift in the fluorescence. How the chromatic shift occurs is explained by changes in the cellular concentration of H342. As H moves across the cellular membrane because of the ABCG2 activity, the blue and red fluorescence ratio follows a curve as shown in typical H red versus blue dot plots. In addition, sample manipulation heavily affects site population resolution, and multiple efforts should be addressed towards minimizing the sample processing time. Excess time spent on isolation and preparation of cells can result in suboptimal SP analysis. For this reason, samples should be processed immediately, avoiding density gradients if possible. And there is no doubt that different factors may alter the site population distribution from the tip to the G0, G1 cluster. As I told previously, it is generally accepted that H low are more primitive site population cells. However, and in many situations, specimens assigned for the site population have a low cell number of SP cells showing heterogeneous profiles. We believe that this method can be difficult for most investigators, first of all because the ability to discriminate site population cells is based on the differential retention of H dye or dicycled violet dye during a functional assay. Second, because of the difficulties in setting the right experimental conditions. And third, because the analysis of the acquired data requires an extensive expertise on flow cytometry to accurately detect the site population events. The prevalence of SP cells is very low, 0.05% of normal whole bone marrow in the mouse, and 0.9% in normal human samples. However, it should not be difficult to find the set population if the whole procedure is followed as we have described. And here you can see an animation showing the whole staining process from 0 to 2 hours, the time incubation needed to unveil the site population tail in human specimens. And this also shows the whole staining process from 0 to 2 hours, the time incubation needed to unveil the site population in human specimens. Since mutant ABCB1 and other transporters, known or unknown, will be able to efflux Hedge or dicycle violet, cells clustering within the site population may be wrongly considered as primitive stem cells, especially when such transporters are expressed in cancer and leukemia. Although the identification of the site population is based on Hedge or dicycle violet staining, alternative assays to examine the site population in cancer samples are needed, such as co-staining with a monoclonal antibody against the external domain of ABCG2. Although we were initially interested in the therapeutic application of SP cells, we moved forward to try to enlighten their role in cancer. Site population cells exist in tumors and could have a key role in drug resistance and, of course, also be able to evade chemotherapy. And then we look at the site population in cancer samples. In normal human bone marrow, the site population is composed of very rare cells, which account for the 0.5% of all nucleated cells, or less. And however, in bone marrow preparations from leukemia patients, site population cells can be found in large numbers, up to the 15% and more. Here we can see the site population pattern of Hedge efflux in the bone marrow of a patient with acute myeloid leukemia at diagnosis. The site population contains increased levels of site population cells from high to low fluorescence, giving duplicated SP tails and SP profiles. And in this slide we can see the DNA content through high-resolution cell cycle analysis corresponding to the same patient diagnosis with AML, showing a secondary tetraploid subpopulation as calculated from the linear relationship between DNA content and propylium iodide fluorescence. From our experience, circulating SP cells are usually detectable in peripheral blood, and in this slide we can report the only patient we have observed with circulating SP cells in peripheral blood, showing that only 11 SP cells from a total of 100,000 cells in the sample aberrantly express the CD34 antigen, suggesting that they could have a key role in sustaining leukemic cells. The mechanism underlying drug resistance are still poorly understood, but various stem cells often express higher level of drug resistance proteins such as ABCG2 and ABCB1 transporters. Augmented levels of these in cancer stem cells may contribute to the refractoriness of metastatic cancer to chemotherapy and should be considered as new target for drugs. Then we went back to our animal models trying to identify site population cells in human prostate cancers. However, all the efforts to detect site population cells in these tumors were unsuccessful. But surprisingly, the analysis of mice bone marrow cells obtained by flushing the ephemera showed cellular types not present in normal bone marrow. In this slide we can see the different cell morphology in primary bone marrow cultures. And in order to identify these cells, then we prepared cytospins or cytocentrifugations that were incubated with a human anti-cytokeratin egg and analyzed by fluorescence microscopy, showing that the observed abnormal cells were human infiltrating cells. Moreover, then we resorted to analyze normal mice bone marrow samples using the site population assay as well as bone marrow cell from xenografts, showing that almost, if not all, human infiltrating cells were site population cells, suggesting that SP cells may have a key role in bone marrow infiltration, and that bone marrow could be also the reservoir for tumor SP cells. More recently, we are trying to unveil the site population compartment, and we are performing a series of experiments aimed to analyze the microRNA expression profile in SP cells. A large number of microRNAs, up to 108, seems to be heavily correlated in site population cells, and we hope to identify new candidate microRNAs that could play a key role to explain the complex biology of these cells. In addition, we have moved forward more ambitiously to study the site population compartment, and we are performing a series of experiments aimed to analyze the gene expression profile in SP cells. A large number of cells, up to 15,000, are expressed in SP cells, and we hope to identify new candidate genes that could play a key role to explain the complex biology of these cells. In this slide is displayed the gene expression profile of primary tumor cells under hypoxic and normoxic conditions, and the next one displays the gene expression profile of site population cells isolated by means of cell sorting from primary cells. For gene expression studies, primary cells are stained with Hedgehog as described, and keep in mind that site population cells can be initially detected in low numbers, in a small percentage, less than 1%, prior to cell sorting experiments. Subsequently, cells can be resorted to obtain enriched cell cultures in site population cells up to 80%. For gene expression profiling comparisons, the same number of site population and non-site population cells should be sorted. In this slide, data from 2 representative experiments are reported as the mean of triplicate determination plus minus standard deviation. Real-time PCR of mRNA from human astrocytoma cells and gliomas using specific primers for ABCG2, ESMO, and patch in unselected cells is compared with the following slide, showing the same analysis in the site population isolated from the same cells. ABCG2 was inversely expressed when compared with ESMO and patch, lending support to the hypothesis that ABCG2 regulates small levels, whereas the decline in the expression of patch may represent a compensatory mechanism as a result of ESMO downregulation. The relative numbers of initially detected SP cells ranged from near zero to the 3% of the PI propylium negative cells within the live gate. Subsequently, cells were sorted to obtain enriched products in SP cells ranging from 40 to 80%. Accordingly, for the enrichment or purification of SP cells, a representative experiment using hemimembrane astrocytoma cells is shown. SP cells were initially detected in low numbers in a small percentage, less than 1%, prior to cell sorting experiments. Isolated SP cells were cultured for 2 weeks following sorting under the same conditions as unsorted cells, giving approximately 5% of SP cells. Sorted cells were cultured again for 2 weeks, giving approximately 30% of SP cells, and then we resorted to the same experimentation to obtain highly purified concentration of products with SP cells. For competitive experiments using cyclopamine, SP cells were sorted again. Only cell cultures with a minimum of 40% of SP cells were prepared in order to study the effect of cyclopamine and temozolomide on Hedgehog uptake. The significant decrease of SP cells was a consequence of adding cyclopamine, whereas temozolomide had no effect on Hedgehog flux, indicating that the decrease in the site population cells was not a consequence of the depletion of the site population compartment, but also a direct consequence of the molecular mechanisms involved in the affinity of the cyclopamine and Hedgehog dye for ABCG2 transporter. Hedgehog signaling plays a role in many processes during embryonic development and remains active in the adult, where it is involved in the maintenance of the stem cell compartment and also in the stem cell populations. Aberrant hedgehog signaling in some cases can lead to certain forms of cancer. And in the absence of ligand, patch represses SMO, preventing the activation of hedgehog signaling. In the presence of hedgehog, the inhibitory effects of patch on SMO are relieved, and SMO becomes phosphorylated and Gli enters the nucleus to induce the transcription of hedgehog target genes, as you can see in this animation. In summary, patch, SMO, and Gli have been identified in hedgehog signaling cascade, playing a key role in the control of stem cell proliferation. Moreover, aberrant ablation of the hedgehog pathway has been associated with different cancers. Here we propose a model for hedgehog regulation in light of our data with the unexpected role of ABCG2. SMO activity can be modulated by synthetic molecules, such as cyclopamine, and endogenous metabolites. Our results suggest that ABCG2 is directly acting as a firewall on SMO and maybe also acting on other potential targets involved in hedgehog signaling cascade, preventing their binding in a concentration-dependent manner and adding more complexity to hedgehog regulation. And for additional information and for more details on stem cells use and functional drug-resistant analysis, please kindly consult it at the current protocols in cytometry. Thank you very much for your attention. And if you have any questions, any further questions, please do not hesitate to ask. Thank you very much.
All right. Well, thank you so much, Dr. Petriz. So let's go ahead to the question and answer segment. So if you haven't yet submitted a question for Dr. Petriz, now is the time to do so by clicking on the Ask a Question box at the bottom of your screen. So let's see what questions have come in so far. Starting out here, one question. Dr. Petriz, you ready?
Yeah, I'm ready.
Great. First question, which ABC transporter inhibitor do you prefer to use for side population?
Yeah, I prefer the fumitremor, which is a well-reported and a lot of publications in the literature are using this inhibitor. In addition, verapamil is one of the preferred inhibitors, but in my opinion, the better inhibitor is fumitremor, and verapamil is also a good inhibitor for the P-glycoprotein.
Okay. Next question is dye exclusion the only technique that can be used to detect SP cells?
Yeah, dye exclusion is the only available technique in combination with functional flow cytometry, because we are measuring or we are looking at the functional activity of the ABCG2 transporter, and this is fundamental. We need to use functional flow cytometry to identify the site population cells, and in addition, we can combine this functional study with a monoclonal antibody against an external domain of the ABCG2 transporter to confirm that we are looking at true site population cells.
Okay, this next question actually has two parts. I'll read the whole thing and you can answer it how you like. Can we assess the efficacy of drugs on CSC activity? And how can you differentiate between CSC and normal SC?
Yeah, this is a very nice an excellent question, and it is really difficult to identify stem cells. Research has shown that cancer cells are not all the same, and with a malignant tumor or among the circulating cancer cells of a leukemia, there can be a variety of cell types. The stem cell theory of cancer proposes that among all cancer cells, euphoeactus stem cells that they reproduce themselves and sustain the cancer, much like normal stem cells normally renew and sustain our organs and niches. And this view is very difficult to distinguish these different populations. And I think that we should look at the tissue that where these cells are originated, and then we need to look to specific markers, because these cells can be very plastic and can express different markers. And we have very nice results that confirm this, which was done with site populations from brain, showing that they can simultaneously express primitive markers in combination with markers from mature neurons. Yeah.
Okay. The next question we have: is there any role for Notch in the regulation of ABCG2 expression and activation?
Yeah, we have preliminary data, and I've didn't show these slides because we have the suspicion that ABCG2 is also regulating a different signaling pathways that are crucial for the maintenance of the stemness of very primitive progenitors. In addition, we have very nice data about gene expression analysis showing that the most primitive progenitors express high levels of multiple multidrug transporters, and we believe that the most primitive stem cell is associated with a classical overexpression of multiple multidrug transporters, and during differentiation, these cells will lose, and they will have a lack on the expression of these multidrug transporters, with the exception of a minimal amount of mature cells, such in the hematopoietic system. The T cell subpopulation can express the P-glycoprotein, and NK cells can also express multidrug resistant transporters.
Okay, on to the next question then. Have other ABCB transporters been linked specifically to tumor stem cells?
I think that there are in the near future, I think that we will be able to identify new multidrug transporters which are associated with the stem cell phenotype, but also with the cancer stem or the leukemic stem cell phenotype. At the present time, ABCG2 is a very promising marker to identify very primitive progenitors, and we need more data to learn more, not about the phenotype, because there is a classical or a growing knowledge to study the stem cell compartment based on the functional studies and not only the phenotype. I think that most of stem cell researchers will agree that it will be very difficult to associate a combination of markers able to identify the stem cell phenotype, and in addition, we are also looking at the normal stem cell phenotype, for instance, for the hematopoietic stem cell system.
Okay, on to the next question. Is the side population necessary or sufficient for a cancer stem cell phenotype?
No, because there are some cancer stem cells which lack the SP phenotype. They have a different patterns of markers or a different phenotype, or in addition a different functional activity with a minor degree of overlapping, and not always this function or this phenotype is overlapping between different cancer stem cell types.
Okay. This next question seems very specific, so we'll see how it goes. Will there be a significant difference between CD44 positive, CD24 negative cells in circulating tumor cells in blood compared to normal circulation of stem cells in blood?
Yeah. This is also a very nice question, an excellent question. And the main problem is when we look at the circulating tumor cells, is that we are looking at very rare cells. Circulating tumor cells I think that the concentration of these cells can be very low. But if we look at the circulating cancer stem cells, we will have only 1 cell per liter of blood, and this is limits. And this is very difficult to truly identify the phenotype of these cells. And in addition, I've observed for very different tumors that these cells can express aberrantly different markers, and this is fundamental to a study astrocytomas or gliomas, which they can lack the CD133 cluster of differentiation marker, and I believe that it will depend of the origin of these cells, and in addition, because they can aberrantly express multiple markers, and it is difficult to associate a specific stem cell marker with a cancer, because we also lack a specific marker to identify leukemic stem cells or cancer stem cells. And I believe that we need to look at the combination of phenotype and function, and go deeper with more functional studies, mainly by using multiparametric flow cytometry.
All right. The next question we have you experienced changes in efflux capacity in the cells if the cells remain in culture for an extended period of time?
Yeah. This is also a very nice question, and we also have observed that during cell culture, cells undergo differentiation, and they will lack the expression of the multidrug transporters. This is mainly observed under concentrations of oxygen ranging the 21%, this is the atmospheric concentration, and if you culture your cells under low oxygen concentrations, you will be able to maintain the SP phenotype, but also the activity. But if you would like to preserve at the maximum the activity of the multidrug transporter, you can resort to use very low concentrations of some drugs or that will help to maintain the expression of the multidrug transporter. But this is true. There is a decrease on the activity, but also on the expression of this multidrug transporter.
Okay, on to the next question. Does ABCG2 transfection drive or confer the SP phenotype?
No, this is also a very nice question, because most of people during multiple talks or during when we perform transfection experiments are asking me about the possibility to transfer the stem cell phenotype, but it is not true. We will transfer only I'm sorry. We will transfer, or this transfection will drive only the multidrug resistance capacity of these cells, and we need more additional modifications in these cells to confer a stem cell phenotype.
Okay, on to the next question. Is there any physiological function for ABCG2 in normal stem cells?
Yeah. In fact, we believe that the physiological function in very primitive, normal or healthy stem cells is to protect the stemness of this compartment, and we believe that these transporters act like a firewall, trying to protect transaction signaling aimed to conserve the stemness of these cells. In addition, and this was reported by Zhu and collaborators and published in the Nature Medicine, because they were able to identify that the expression of the ABCG22 transporter in very primitive hematopoietic stem cells, it seems to repress the hematopoietic system, and this will allow to maintain the stemness of this compartment. And in addition, the ABCG2 transporter will protect these cells against xenobiotic compounds or other toxic molecules that could kill these cells.
All right. It is now time to wrap up the question and answer session. So I'd like to let you know that today's webinar has been recorded and it will be available for viewing in the next few days. We will send you an email with details on how to access the recorded webinar, along with instructions on how to personalize and print a certificate of attendance, as well as download a PDF of the slides. So on behalf of today's speaker, Dr. Jordi Petriz, and our sponsor, Thermo Fisher Scientific, we greatly appreciate your attending today's webinar, and we look forward to your attendance at future events from Current Protocols.